10th Biology, Unit 16, Plant and Animal Hormones. This is a new lesson. So in this part, let us discuss about the introduction of the lesson and the first plant hormone, auxins. What are hormones? Hormones are chemical substances. They are present in plants and animals. They are signaling molecules. They carry chemical signals. Because of the reason, the hormones are called as chemical messengers. In the previous lesson, we learn about nerve impulse. Nerve cells carry electrical signal. It is a message. Here the hormones carry chemical signals, so they are called as chemical messengers. These chemical messengers control physiological activities of plants and animals. Plant hormones are called as phytohormones. In plants, the cells are capable of producing hormones. Many cells are capable of producing hormones. Whereas in animals, the hormones are produced by glands called as endocrine glands. These hormones of plants control growth of the plant, root development of the plant, flowering of the plant, fruit formation, ripening of fruit, etc. In animals, these hormones are produced by endocrine glands. Endocrine glands are found in various regions of our body. Already you have learned about endocrine glands in 8th standard. Some of the endocrine glands are known to you. Pituitary gland, it is found inside the brain. Thyroid gland, found at the neck region. Pancreas, it is attached with the digestive system. And the pancreas, hormone of pancreas controls blood sugar level. So these endocrine glands are found in various parts of the body and they produce hormones, animal hormones. In animals, the hormones are transported by blood. Hormones are carried by blood to various regions of the body. So in this part, the beginning of the lesson, in this lesson at the beginning, plant hormones are given. So let us start with plant hormone. There are 5 types of plant hormones. They are auxins, cytokinins, gibberellins, abscisic acid and ethylene. Of the 5 plant hormones, the first 3 influence growth. They are growth promoters. They promote plant growth. Abscisic acid and ethylene, they inhibit plant growth. Abscisic acid and ethylene are growth inhibitors. The first 3 hormones are growth promoters. Auxins were the first plant hormones discovered. Auxins were discovered first. They are produced at the tip of the root and tip of the stem. Auxins are produced at the tip of the stem and tip of the roots. From the tip of the root and stems, the auxins spread to other regions. Auxins move to other regions where they show the activities. They control physiological activities of the plants. When we see the discovery of auxin, we have to learn Charles Darwin's observation. Charles Darwin is a famous scientist. He is an evolutionist. he observed a bending growth in canary grass. When the tip of the grass is undamaged, when the tip is intact, the plant grows and the plant bends towards one side, single side bending towards a light source. If the tip is removed in this plant, the tip is removed, there is no growth, there is no bending growth. And he concluded that some substances are produced at the tip of the plant. These substances spread towards the base and cause the growth of the plant. Here tip is not there, so there is no substance produced at the tip. There is no substance to promote the growth. Charles Darwin observed unilateral growth and curvature. Uni means single, lateral means side, curvature bending in canary grass, coleoptile. Coleoptile is a covering found around the tip of the young monocot plant. When a monocot plant comes out of the seed, the tip is covered by coleoptile. And Charles Darwin came to the conclusion that some influence was transmitted from the tip of the coleoptile to the basal region. Some chemicals were transmitted from the tip to basal region. That results in growth. Later, Wendt, a scientist, he identified the substance responsible for the growth as auxin. Then the substance responsible for the growth bending movement was auxin. That was discovered by Wendt. Now let us see Wendt experiment. Wendt conducted experiment on Avena plant. Avena is a scientific name of oats. You know oats very well. Oats is available in all the shops. This is a seed of oats or Avena seed, and this is coleoptile. When seed germinates, the young shoot system comes out. That is covered by a covering called as coleoptile. This is seen in monocot plants. This is coleoptile. Inside the coleoptile, a young leaf is found. What did he do? He cut the tip of the coleoptile. He removed the tip of Avena coleoptile. Now tip is not there. After the removal of tip, there was no growth. In a second experiment, he placed the agar blocks on the decapitated coleoptile tips. Decapitated tips means the cut tips. Decapitation, removal of the tips. After the removal of the tips, agar blocks. Agar is a gel-like substance. A small piece of agar was kept on the tip. Now agar block was kept on the tip. Now also there was no growth. The coleoptile tips didn't show any response. There was no growth after keeping agar block on the tip also, after cutting the tip. Now let us see his third experiment. He removed the tip of coleoptile and placed the removed tip on agar block. The tip was removed and it was kept on agar block. If there is chemical in the tip, the chemical will spread into the agar block. That is possible. Then after 1 hour, he removed the agar coleoptile tip from the agar block, and agar block was kept on the tip. After an hour, he discarded the tips and placed this agar block on the decapitated coleoptile. Cut end of the coleoptile, he kept the agar block, not the tip. Agar block alone was kept. Agar block was kept. Now there was growth. He found growth. What may be the reason? The tip produced some chemical that chemical spread into the agar block. When we keep this agar block on the cut end, this chemical from the agar block flows downwards and promoted the growth. On keeping this agar block, the plant started growing. So this chemical was auxin. From this experiment, when concluded that chemical diffusing from the tip of the coleoptile was responsible for the growth, and he named this chemical as auxin. The meaning of the auxin is to grow. There are two kinds of auxins. Natural auxins are chemicals produced by the plant. Synthetic auxins are artificially prepared by man. IAA is an example for natural auxin. IAA, indole 3-acetic acid. Indole 3-acetic acid is natural auxin. 2,4-D, 2,4-dichlorophenoxyacetic acid is synthetic auxin. There are some other natural and synthetic auxins are there. PAA, phenyl acetic acid is natural auxin. IAN is natural. IBA is synthetic. NAA, naphthalene acetic acid is synthetic. 2,4,5-T is synthetic auxins. Auxins are responsible for growth of the plant. Now let us discuss the physiological effects of auxins. Auxins promote growth. Auxins are produced at the tip of the plant. Already we have learned from the experiment. In the previous class, 9th standard, we have learned about apical meristem, and in 10th standard also we learned about apical meristem in the first lesson, biology lesson first. The apical meristem undergoes cell division, so number of cells increases in this apical meristem, and auxin produced at the tip of the plant spreads downwards and control cell elongation. Just below the apical meristem, because of the auxin, the cells become longer. That is cell elongation. Auxins influence cell elongation. As a result of this, the stem grows towards the light. When the light comes from the side, the plant shows bending movement. More auxin spreads towards this area, so here cells are more longer, that results in bending towards the light. Auxins promote the elongation of stems and coleoptiles, which make them grow. Auxins causes elongation of stem and coleoptile. Auxins promote root formation in low concentration. When the auxin concentration is high, it inhibits root formation. At low concentration, auxins promote root formation. Auxins induce root formation at low concentration and inhibit it at higher concentration. This is apical bud. What is apical bud? A bud present at the tip of the plant is apical bud. The black color buds are lateral buds. Lateral means side. The lateral buds grow sideward, towards the side. If the apical bud is present, the auxins produced by the apical bud flows downwards and suppress or stop the growth of lateral buds. If the apical bud is removed, here the apical bud is removed, now there is no auxin is produced. In this time, the lateral buds grow well. If the tip is removed, lateral buds grow. If the tip is not removed, the auxin present in the tip moves downwards and suppress the growth of lateral buds. This is called as apical dominance. This is an important term. Auxins produced by the tip apical buds suppress growth of lateral buds. This is called apical dominance. Auxins are used to produce seedless fruits. This is seedless watermelon, seedless lemon, seedless grapes. So seedless fruits are called as parthenocarpic fruits. They are produced without fertilization. By spraying auxins on the flowers produce seedless fruits. Seedless fruits without fertilization are induced by the external application of auxins. Example, seedless watermelon, grapes, lime, etc. Abscission is a process of falling of fruits, falling of leaves, falling of flowers. After maturation, the leaves fall down, flower fall down. This is abscission. If auxin is low level, the young leaf, young flower, young fruits also fall down. If auxin is present, the young leaves do not fall down, young fruits do not fall down. So auxins prevent abscission. Auxins prevent the formation of abscission layer. What is abscission layer? At the base of the fruit or leaves, loosely arranged cells are formed. That is called as abscission layer. Auxin prevents the abscission layer. So auxin prevents the falling of leaves, flowers, fruits before maturation. Auxins prevent abscission layer. Auxins are responsible for formation of seedless fruits. In the next video, we shall discuss about the next two hormones.